Hey guys, this will be our last video for Unit 9, and I'm going to just talk about this really briefly to talk about some of the common laboratory tests that we can carry out when we take a sample of blood. Now generally when we take a sample of blood, the first thing we'll do is a complete blood count, or it's often called a CBC. When we do a complete cell count, we're looking at all of these things that are listed here. So we're counting the number of red blood cells, we're counting the number of white blood cells, we also do a differential white blood cell count, which is going to be the number of the types of white blood cells. So we want to know that as well. We count platelets, and then we look at hemoglobin value, so how efficient are your red blood cells at transporting oxygen, and we refer that by the amount of hemoglobin in your blood. And then we also do a hematocrit count. Now hematocrit is the percentage of red blood cells relative to the whole blood cells. So how many red blood cells are there compared to white blood cells, platelets, and plasma. So that's a typical red blood count. That kind of takes stock of everything we can find initially from a blood sample. So hematocrit is measured specifically as milliliters of red blood cell per 100 milliliters of whole blood. So what we're doing is we're looking to find that number of red blood cells relatively in your blood. Remember, we want to have around 4.5 to 5 million red blood cells per microliter, and then of course we can use that number to scale up so we know that your red blood cells is in a normal or healthy range. Now I'm not too worried about you guys knowing the ranges for that, just know the definition of a hematocrit. Hematocrit's easily taken by putting a whole blood sample in a centrifuge, and we spin it around for a known amount of time for a certain speed, or RPM, rounds per minute, or revolutions per minute, and that will give us that will separate the red blood cells from the rest of the sample, and we'll be able to take our hematocrit measurement. So here's a look at hematocrit studied in the test tube. Right here we're going to have our normal hematocrit value, and that we talked about before. Red blood cells account for about 45% of the formed elements, 45% of the whole sample. And then we get different values. So for anemia, that's of course where we have low red blood cells. You can see the value of red blood cells in comparison to the rest of the blood. So that shows you that the person has anemia. If we look over here at polycythemia, this is an example of a blood cancer where your red bone marrow produces way too many red blood cells. So the number of red blood cells is drastically up compared to the whole blood sample. And this can cause a bunch of complications, including clots and other things that are, relatively dangerous. Now this example right here on the end is quite remarkable. In the case of dehydration, you can see the volume, the whole volume of blood, everything else is up here, the whole volume of blood is decreased. Okay, the plasma has gone down, but the red blood cells stay about the same as a normal sample. So of course that means the red blood cells are going to make up a bigger proportion of the whole blood sample. So that's not going to be a good thing. So dehydration is something that we should always be very, very careful about. Hemoglobin tests. Now when we're measuring hemoglobin, we're looking at that protein that's in the red blood cells that carry the oxygen, and we're looking at it as measured of grams per hemoglobin per 100 milliliters of whole blood. We perform that test by using a process called electrophoresis, and electrophoresis is when we pass a current through a sample, and if you've ever seen a DNA sample on a crime show, for example, where they get the bands of the DNA separating, that's the product of electrophoresis, and we do the same thing to find hemoglobin. Red blood cell counts, we talked about these numbers, blood cell counts, we talked about these numbers as we went through each formed element. So the red cell count should be between 4.5 to 5.5 million. White blood cells should be around 5,000 to 10,000 cells, and platelets should be 150 to 450,000. And now all of these, of course, are going to be per microliter. Okay, so we always keep the units so that we know what we're comparing. So when we're talking about blood cell counts, it's generally millions of cells or thousands of cells per microliter. A blood smear. Now, blood smear is when we take our blood sample and we smear it on a slide so that we can look at it under a microscope. Okay, so when you guys looked at blood under a microscope in the lab, that was a blood smear. So from that we can look at the red cells, we can see platelets, we can perform a differential white blood cell count because we can see the different types of white blood cells. And one of the other thing that we can also do is we can see parasites. Quite often, if a parasite's present in the body, it could be present in the blood. And if you put blood on a sample and you're looking through it and all of a sudden you see something that shouldn't be there but still alive or was alive, then you have a case of a parasite. So that's a pretty interesting slide to look at. And finally, these are all the things that show up in the blood, generally in the plasma. Electrolytes, glucose, nitrogenous waste products, creatine, enzymes, lipids, plasma proteins, hormones, of course we talked about, vitamins, antibodies, which are part of our immune system, and drugs. All of these show up in blood chemistry tests. So there's various different tests we can perform to find out the levels of all of these products. And when we compare that to what we know are normals, and then we can tell if there's an imbalance or if there's a problem with this particular sample. So I'm not going to go into how these tests are performed. Just know that we can test the blood for a lot of different things besides just the formed elements and the volume of plasma. Another thing we can perform with blood are coagulation studies. Now these are important because they let us know if the blood is capable of healing itself healthy and carrying out hemostasis. So very often before you have a surgery, for example, doctors need to know if your blood is efficient at stopping bleeding or clotting, so that they know when they make an incision, or if you bleed, they know what to expect. So when we do these coagulation studies, we're looking for those clotting factors that can be present in the blood. We're also looking for how long particular blood will stay fluid before it turns into a solid. So we measure the clotting time. Now don't worry about these abbreviations, PT or PTT. We also look at capillary strength. Are capillaries are they weak? Are they subject to bleeding if they're disturbed? And we'll also look at platelet function. So these are all types of studies, but they're all linked to finding out if the blood is healing normally to prevent bleeding. And then finally we'll look at the tissue that actually makes our blood cells, the bone marrow. And it's common to get a test if there's problems or issues with the blood. They'll often take a biopsy of the bone marrow to find out what's going on. Now a biopsy is a small sample of a tissue. The way they do this is they take a needle and they put it either in the sternum, the bone in the center of the chest, the sacrum, which is a part of the hip bone, or the iliac crest, which is also part of the hip bone. So this type of sample would be how we'd use to diagnose a lot of bone marrow disorders, which of course will be linked to bone disorders or blood disorders. So leukemia, some types of anemia, this is the test that we would use to diagnose those conditions. Now, if there's damage or if there's a disease with the bone marrow, how would this affect the blood? So that's a pretty interesting question to ask. If the tissue that produces the blood is damaged, how will that affect the blood in that patient? Will that patient stay healthy, or will there be issues? And then if you think about this even further, what would be some of the side effects, or what would be other effects in the body if the tissue that produces the blood is damaged? So we could look at things like bleeding, we could look at things like anemia. So there's a lot of repercussions for issues with our red bone marrow, because of course our red blood cells, or our blood in whole in general, is so important for circulation and bringing things around the body. So what would happen as a result of damage to the bone marrow is a really interesting question, and if we had a lot more time in this course, we would spend some more time on that. Okay guys, thanks a lot.